Trisha here again. We're going to be going over a kind of meaty worksheet for today. So we're just going to go ahead and dive right on into this. Let me go ahead and exchange this scene for this one, and hopefully you guys can see the screen. This is a long block of text that we can just go ahead and skip through. Main thing I want you to focus on here is that it's saying consider the two signaling systems and their associated vocabulary below. You have on the left something here called your G protein receptor system, and then on the right you have your receptor tyrosine kinase system. And the first question here is asking, how are these two systems similar? Well, main thing with both of these is that they are receptors, and you can even see it in the name here. There's a receptor word, there's a receptor word, and what does that mean? Is that it means that both of these receptor proteins are first bound in the cell's membrane. That's the first thing. And if you want, I'll just do a little dot here so that way we can kind of like bullet point it out. Second thing you want to add here is that binding of signal molecules to receptors activates them. Then the last thing you're going to add here is that these activated receptors interact with inactive relay proteins and therefore activate them. Pretty straightforward there. So now we'll move on into how are they different. So I'm going to create a little bit of a line here in this box. So that way, and I'll just make that a little nicer looking, right about there. I'm a stickler for things looking slightly pretty. Okay, so we're gonna put over here in blue color, the G protein. And then on the right in magenta, we'll say for the TKR. So that's the tyrosine kinase receptors. So the main thing I want you to walk away with in terms of how they are different for the G protein is that it becomes functional. Oops, that doesn't look right. When activated by its signal molecule. Another thing about the G protein is that, let me make a little dash here, and if you want, I'll just zoom in a little bit so that's a little bit easier to read, since I think it's a little thin on my end, is that it is activated by a specific enzyme which produces cell response. And then the last thing I'm going to add down here is a little bit of like a tie-in to OChem, just a tiny bit, because what happens here with the G protein receptor system is that a conformational change occurs. Therefore, it's structurally different. Without going into like too many details, especially for those watching this who have not taken OChem yet, there's a difference between a conformer and then a constitutional or structural type of change or isomer. So the short way I can explain it is that a constitutional isomer, so like some sort of molecule, it's the same weight, but the weight that it is joined by its atoms, their bonds, are basically different. So that's why it's like I can have the same pentane, but the way that they're joined is totally different, from like N-pentane to isopentane to whatever type of pentane. Meanwhile, conformational is everything is the same, but the way that it's rotated, including the way that the way that things are connected, are the same. But the biggest difference is that it conformationally changes, and therefore where they're actually attracted in terms of their bonds. So like if they're eclipsed, so that means their bonds are too close to each other and therefore they're not happy. Or if they're staggered and they're far away from each other, that changes on how you rotate around the bond itself. And that takes a lot of like looking at things in 3D, and it's a little complicated, but the more you do it, the easier it gets. So that's just something important about G protein. Again, going back to what is good, not good about, but what Dr. Ray talks about with bio, which is structure function. If you change the structure, you change the function. And as you learn in OChem, that it's sometimes like it's not just the structure, you're changing the way that even the structure rotates and things like that. Anyway, moving on into the tyrosine kinase system. Tyrosine kinase receptors. You need two of these TKRs to activate and then you also are going to need, they're just not membrane bound compared to your G proteins. Another thing is that these TKRs can activate up to 10 different, I'm just going to shorten that, specific, make that go up there, specific relay proteins inside the cell. And produce multiple responses. All right. So we're done with that one. Moving on to number 3. It asks here, both systems can generate elaborate multi-step signal transduction pathways. These pathways can greatly amplify the cell's response to a signal. The more steps in the pathway, the greater the amplification of the signal. Explain how this amplification can occur. We're going to switch to a black font again. And basically, and I'm going to abbreviate this just to make it a little bit easier, and hopefully you don't hear my dogs because my puppy is extremely loud. He just wants attention. So in signal transduction pathway, each second messenger can catalyze more than one reaction. And each reaction, produces, not produces, that's like the wrong word. Each reaction can potentially trigger not more reactions, I already said that. They can trigger effects produced by a single signal molecule. There you go. Moving on. Now we're going to go into what is mitosis. Let's see, hopefully we got, oh, look at all the people. Hi, everybody. If anybody wants to participate and just say, somebody tell me what mitosis is really quick. I'll give you a little bit to answer. I can also have time to take a break and drink a little bit of some tea. Anyone want to answer that? Anyone at all? As I move back and forth, like the ham that I am. Nope. That's okay. We can go ahead and move on. Get to the end of this so you guys can go back to studying. I know what you're here for. I'm here. I know you need to study. You also want those extra credit points. I got it. So, overall purpose of mitosis, bullet point, produce daughter, and hopefully my writing is not terrible, cells. Identical to the parent cell. That was terrible. And then another bullet point to do this. Cells must first duplicate all chromosomes, then equally divide them among the daughter cells. And that way, so each has the same, as I scoot this over to the side since I realized I actually had a lot more room to write with, and they have the same number and kinds of chromosomes. As the parent cell. Okay, that's all you have to write down for mitosis. Now, for the next one, just gonna go to the next page. And again, let me know if you want me to go back and you guys can read some of this. But again, sorry if I haven't been posting. I haven't been. I realized I kind of messed up, but going forward, my videos will be posted the day of after the live stream on the Learning Center YouTube channel. I fixed it. Yay me. Okay, so what this is asking here on the next page, and you're going to see a bunch of pictures of a cell, is that it's asking, you know, draw what you would expect to see if you were looking at a cell in a stage of mitosis. So again, this is all about highlighting here in, let's make it a nice bright, somewhat bright blue. Mitosis is what we're talking about. Mitosis, not meiosis or meiosis. It's the same word. Choose the one that makes most sense to your brain. I go meiosis because it makes me remember, oh, it's not the same. Look at the cell stage in mitosis indicated. If no circle is present, draw what you would expect to see at a given stage. Pretty much on this page, everything has been drawn for you, quite frankly. So what I want you to write down here is basically what you see for each of these phases for mitosis. Not going to take long. It's just little words to kind of practice what you see in mitosis. So what you're going to write down here is that the first thing that you see in prophase, so I do it in blue, okay, and I'll have a little arrow pointing here, and all you're going to say is that the nuclear membrane disappears. Then the next thing that happens here in the chromosomes for prometaphase, and I'm just going to zoom in a little bit here, notice how there is something missing here. You have 1, 2, 3, 4, 5, 6. You have 6 in prometaphase. But how many did you have over here in prophase? 1, 2, 3, 4, 5, 6, 7, 8. So there was something to draw here. So all you're going to do is just draw 1. Oops, got to have them touch. 2, 3, 4. Just draw those 2 extra chromosomes that were missing in prometaphase. And then I'll have a little arrow. And then all you're going to say here is that the chromosomes begin to align at equator, aka the metaphase plate. All right. Then we scoot on down here to metaphase. And then I'm just going to draw a line in between all of these chromosomes here. Then all I say is, here's an arrow, aligned at equator. And that this over here, just so you practice, this is a microtubule. They're not 2 words, it's 1 word. Then what happens over here is the following. I have to turn my iPad around to make sure this looks good. And then, sorry. That looks terrible, but I digress. And this is where you say that sister chromatids split at opposite poles of the cell. So they're opposite poles. This is one pole over here. This is one pole over here. Okay? Next one, telophase. And I'm just going to draw a squiggly line around this one, squiggly line around this one, because essentially what happens is that it's the reappearance of nuclear membrane. Perfect. Cytokinesis. I'm going to draw another squiggly line right here. I'll draw an arrow. And essentially what's happening here in cytokinesis, which I feel like is forgotten sometimes, including by yours truly, in mitosis and in meiosis, is that this is the cytoplasm division that gives away, it's giving 2 cells. And then you have what's left over at the end of mitosis, which are your daughter cells that is in interphase before they go into mitosis. And just to go over this, because you will have this on your exam tomorrow, I'm going to make a circle here. You don't have to write this down, but it's just for your memorization. You're going to have one line here, one line here, one line here. You have the phases of the cell cycle. So you have to know that basically everything in G1, G2, and S phase is basically going to be interphase. And then there's one little section, it's going to be your, like, that's, oops, that right here is basically like, this is where mitosis lives. This is M phase. This is your mitosis. And everything else, that is interphase. And again, you're going to have to remember G1, G2, and S cycle. And remember where they're placed in this area here of the cell cycle. So if you guys want to go over this, just like tell me in the chat. Otherwise, we're going to move on and get the rest of this worksheet done. Okay? So, next page. What are the products of mitosis? Basically, the answer here is pretty simple for the first answer, and it's just basically 2 words. 2 cells. 2 cells. The end. How many kinds of cells are produced at the end of a single mitotic division? 1 kind of cell. And then, so that's a bullet point here, and another bullet point is 2 daughter cells. Oops. And they are identical to each other. And, emphasizing here the and to the parent cell. That gave rise to them. And I'm just going to rewrite that because I don't like the way that I wrote the word gave there. Okay, moving on. 6 centromeres are observed in a prophase cell from another species of insect. How many pairs of chromosomes does this organism contain? Since it says the word 6 centromeres up here, divide it in half. And the reason why I say divide it in half is because you want to think diploid first, that's mitosis. Haploid is meiosis. So half of 6 is going to be 3 pairs. Then for each stage of mitosis, indicate the number of centromeres you would expect to find at the number of copies of chromosomes attached to each centromere. Stage of mitosis, prophase or anaphase. So we'll start with the prophase line, the prophase row. Number of centromeres visible per cell in prophase. They gave it away because you start with a diploid. So 3 times 2 is going to be 6. And then the number of chromosomes attached to each centromere is going to be 2. And then in anaphase, you're going to have 12, and then just 1 when it comes to the number of chromosome copies attached to each centromere. Because in anaphase, it's a diploid. So think of, you started with 3 pairs. 3 pairs are going to jump up times 2. So 3 plus another 3 is going to be 6. But it's double that amount because they're going to split. Because again, that's what happened up here. You had it aligned, and then they split into opposite poles. So do 12 divided by 2, 6. Then it says down here, what is the overall purpose of meiosis? Sorry for things looking a little weird with this guy over here. Oopsies. So, what is the overall purpose of meiosis? Make a bullet point right here. To reduce diploid chromosome. Number by half. So again, we're leaving mitosis and going to meiosis, which is different. We're dealing with gametes here with this one. And this is known as a haploid. Haploid, half. Diploid, 2. Haploid is half of a diploid. That's how I remembered it. And then the other thing here to remember is that haploids contain one member of every pair of chromosomes found in the diploid parent cell. And then in what types of organisms and cells does meiosis occur? Bullet point, occurs in eukaryotes. And then occurs in cells that produce gametes, aka ova or sperm. Perfect. Done with this page. Moving on here. We're back to pictures. Okay? We're going to keep this one pretty simple, but I want to emphasize something here that is a little bit confusing about meiosis versus mitosis. On the exam tomorrow, unless you're watching the recording, you already did this. Unless you're watching the recording tonight, which, thank you. You'll probably see some sort of table, and the table's going to say, you know, here's where we started, here's where we ended. This is meiosis. Where, based on the numbers, are you actually, you know, at? Are you in anaphase 1? Are you in metaphase 1? Are you in anaphase 2? Are you in metaphase 2? And it can get really confusing really quick. You might also find something on the exam tomorrow where it's going to ask you the difference between a chromatid and a chromosome, and then what happens to chromatids in metaphase or anaphase 1 versus metaphase and anaphase 2. You might also find a question that asks you, where does crossing over happen and what is crossing over? So I'm going to make this one very, very short and straightforward as to what I would like for you to know on this page, because it's asking if there's no circle, just like the last one, if there's no circle is present, draw what you would expect to see at the given stage. Everything here is pretty accurate except for one thing. It's right at the end. And can anyone in the chat tell me why there's something missing at the end? And if you don't want to, that's okay. I'll give you guys a little bit of like moments to kind of write something in the chat as to why I'm like, why is there something missing at the bottom? Why would I say that? Actually, I changed that to purple. Okay. Here's why. At the end of, this is over here. I'm going to put this in blue. This is your meiosis 1. And then I'm going to put a line right in the middle. And this over here is meiosis 2. So that's the first thing I want you to know, that there is a difference between the 1 and 2, aside from the fact that the Roman numerals kind of give it away right here. In prophase 1, so I'm going to make an arrow right here. In prophase 1, this is where tetrads form. And a tetrad is basically a group of 4 chromatids. Okay, so if you don't remember that, make sure you remember that. Maybe you can even write it down on this worksheet that you have tetrads form around prophase 1. This is also where crossing over occurs. And the reason why I wanted to just emphasize that is because there is a difference between a chromosome and a chromatid. And I'm going to draw that poorly, mind you, right here on the side. This is, wow, this is really bad. I am so sorry. I'm not an artist, and OChem is making me turn into an artist, and I kind of hate it. This is a chromosome. So this here is a chromosome. But we don't really deal too much with, I mean, we do have chromosomes in meiosis, don't get me wrong. But what we're looking more at in a chromosome or in meiosis is this. So now I'm making a circle. I'm going to make a little dot here. And then I'm going to make 1, 2, that's not too bad, and 3, and 4. This is what we're dealing more with over here. This is the chromatid. Another thing that gets confusing is centromere and centromer, if I remember correctly, but it's centromere. This right here that I'm circling all in yellow, I'm just going to do this. This right here is a centromere. And that the chromatids are attached to it. And then I'm going to draw in different colors here why this is important to know. There's 2 colors here that I use versus up here where it's majority going to be all in red. So a chromatid is really good to understand because in this whole shebang right here with meiosis 1 and meiosis 2, what happens here is that as the nuclear, once you go through prometaphase, which is where the nuclear envelope goes away or disappears, either way, you're then going to go to metaphase over here, right here, okay? And you're going to notice that the tetrads line up. And then they split in anaphase. Because essentially what happened here is that in anaphase 1, the tetrad split. And that you have the chromosome halved, and the chromatids basically took away, are starting to piece away things. They have different colors because this is basically, the red is parent, or the blue is parent 1. And this red is parent 2. It took in crossing over little pieces of one chromosome, little pieces of another, and it created those tetrads. That's how genetic diversity occurs. And then what happened in anaphase 1, after they lined up in metaphase 1, very similar to mitosis. In meiosis, in anaphase, they split, meaning that this here went in half. But if you notice, it has a little bit of parent 1 and a little bit of parent 2. And it's going to stay like that the whole time, where it's mixing it up. It's genetic diversity occurring, which is good. And you'll learn more about that in by 213 as to why. But if you noticed, that's not really what happens with the chromosome. If it splits in half, it's like mirroring each other. You're not really mirroring with a chromatid. They're not like a perfect overlap of each other. They're not like entomers or whatever. It's because they've taken genetic material from one parent, genetic material from another parent, and it's attached to different sites of the chromatid. Those alleles inside of the chromatid are different in each time you're replicating it. And it's going to continue doing that as you go into meiosis 2. But what's the difference between meiosis 1 and meiosis 2 is that meiosis 2, their metaphase and anaphase, and I'm going to put circles around both of these, they mimic mitosis phases. So it's meiosis 1 is where it's different because you're having the chromatids form tetrads. Crossing over occurs in prophase. The nuclear envelope goes away in prometaphase 1, as we say here. The tetrads start to line up at the metaphyseal plate, the equatorial plate, and then they split, the tetrads split in anaphase, which essentially means that this chromatid splits apart. And then that creates these daughter cells down here. These are the 2 daughter cells. Then it goes through it again, and that means after cytokinesis, number 2, this is why I said there was an error here, because there's 2. There should be 1, 2, 3, and 4 daughter cells. That's what was missing, is that we were missing these daughter cells. And then each one of these daughter cells, they're going to have the same number of chromosomes. But it's each has, bring that up, each has half the original amount from the first time it happened in prophase. And the reason why is because these are, again, haploids. They're not diploids. So understanding that will be good when you go into the exam, I hope. And I hope this has been helpful. We're almost done. So really, the reason why I want to do this is because what we're about to go through is just re-emphasizing everything I just talked about. Yay. So we're just going to blast through this, and then you can go back to studying or have a great rest of your day by the time you're watching this later on YouTube. So it asks how many cells are produced at the end of mitosis. It's 4 daughter cells. How many chromosomes and which chromosomes does each of the daughter cells contain? Little dot right here for a bullet point. Half number of chromosomes in each daughter cell, and contain one member of each chromosome pair found in the parent cell. And now we move on to 6 centromeres. All right, let's go to purple. Okay, like magenta purple. 6 centromeres are observed in a prophase 1 cell from another species of insect. How many pairs of chromosomes does this organism contain? So it's going to be 3 because until it goes through the haploid process, it's similar to mitosis. So it's 6 centromeres, divided by 2, it's going to be 3 pairs. Now we start to get into something a little different. Now we're going to talk about the meiosis stage for anaphase 1 versus prophase 2. The number of centromeres visible per cell will be 6 in anaphase 1, which then means the number of chromosome copies attached to each centromere is going to be 2. This is different than what we had up here. Well, not too different. Sorry. I'm skipping ahead to the next one. It's similar, prophase 1, so anaphase 1 and not anaphase, prophase was the same until we got to anaphase. So if you notice the anaphase in mitosis, it's 12-1 for number of centromeres for the number of chromosomes. In anaphase 1 of meiosis, it's 6-2. Okay? Then prophase 2, number of centromeres visible per cell is going to be 3. Number of chromosome copies attached to each centromere is going to be 2. Then we're going to move on and talk about this stuff, which is non-destruction of sex chromosomes during human gamete formation may lead to individuals with sex chromosome trisomy. An individual with the sex chromosome trisomy of XXY may have resulted from non-disjunction occurring in, and we'll go through the list. True or false? Meiosis 1 in the father's sperm production. That's going to be, going to put that as a green color. It's going to be true. Next one will be false for meiosis 2 in the father's sperm production. So meiosis 1 is true in the father's sperm production. And it's because it would produce gametes with both X and Y and some without X or Y. Meiosis 2 in the father's sperm production. And if there was non-disjunction during meiosis 2, it would produce either 2 X's, 2 Y's, or no X or Y. Moving on. Meiosis 1 in the mother's egg production. This one is going to be a true because non-disjunction in meiosis 1 could produce eggs with 2 Y's, oh, not 2 Y's, excuse me. Wrong letter. 2 X's or no X. And for meiosis 2 in the mother's egg production, it's going to be a true because it could produce eggs with 2 X's or no X. And it could also, because if fertilized by a sperm carrying carrying a Y chromosome, a XXY individual could be produced. All right. Now, how do they differ? Again, this is going to be a repeat of pretty much a lot of the stuff that we went over. So, what similarities do you see when it comes to interphase of mitosis and meiosis? So I'm going to write this in blue. And what do they have? Like what similarities they have in interphase? They're identical. Let me move that to the side a little bit. They are identical. In terms of prophase, I'm just going to make this a little bit bigger. In terms of prophase, the chromosomes coil and condense. Spindle forms. And the nuclear membrane or the nuclear envelope breaks down. I'm just going to rewrite the word break. I don't like the way that looked. There. And in metaphase, something always lines up at the equator. I'm just going to make this look a little better. At the equator. Then in anaphase, something always moves to the opposite poles of the spindle. And telophase, two things always happen. Nuclear membrane or envelope reforms, and the spindle breaks down. All right, moving back to let's do a nice green color, because now we'll talk about what differences that we see. First difference that we see here is that there's no DNA dupe in the interface section of meiosis II. There's also another thing that is a little different, is that the G phase is possibly shortened. Because if you remember, you go through G1, and then you have S phase, G2, and then you have the mitosis phase or M phase. It's like, if you do it again a second time, it might be shortened because it's like, yo, we don't have to use as much energy. Like, yeah, just saying. So, prophase, I'm going to write down something and then I'm going to explain it because I just want you to get through this, okay? So, in prophase 1, homologous chromosomes synapse. And now what does that mean is that I'm going to go back a little bit, if it's okay with you guys, because we're right at the end. Like, this is like, oh, we do have that guy. Okay, I'll try and make this quick. Okay, homologous chromosome synapse basically means that in prophase 1, you have parent 1, you have parent 2, and we have the crossing over that happens. And that's what a homologous chromosome synapse means. It means that, you know, as they align side by side and crossing over occurs, it's basically where, you know, stuff from one chromatid, remember I did that in different colors, that the one chromatid from one chromosome exchanges a portion of, another portion of chromatid from its partner that it made in that, you know, tetrad, okay? And there's a physical exchange of material that happens, which again, it leads to that genetic diversity. That's what that phrase means. Homologous chromosome synapse. It's where we have the crossing over occurring. Okay, I made it quick. Yay. Metaphase. Basically, what we have here is that in metaphase 1, the synapt pairs line up And then for this one, anaphase 1, each homologous pair separate to opposite sides. And the last thing in telophase is that chromosomes don't usually uncoil during telophase 1. So if you're noticing a theme, telophase 1 is a little bit of the strange one versus the second phase. Second phase tends to mimic mitosis more. Meiosis 1 is kind of like doing its own thing. All right. So now, the question here that kind of got cut off because it starts on the next page is if the amount of DNA in a somatic cell equals C, C stands for cell, during G1 of interphase, how much DNA is present during each phase of mitosis and meiosis? So let us go down every row here with some different colors. Mitosis, interphase, you have C in G1, 2C in G2. And I'm just going to zoom in a little bit so you can see that better. Then you have your prophase, you have 2C, you have 2C, you have 2C, and you have 2C. In parentheses, cytokinesis reduces the number of C. We'll go to meiosis next. You have C in G1, 2C in G2. And I'm just going to make this as bullet points because if I don't, I'm going to be confused. So it makes my life a little bit easier. Hopefully it does for you guys too. Everything is going to be about the same. You have 2C, 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 and then 2C. And again, cytokinesis reduces number of C. And then our purpley magenta color, meiosis 2, you have C in G1, 2C, oh, not 2C, excuse me. It's just C in G2. And then you have just 1C, 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 1C. Because cytokinesis reduces the amount of C by half C. Okay. And then I believe these are the last two questions, y'all. Last two questions. All right, how do the similarities in prophase of mitosis and meiosis compare to the similarities in telophase of mitosis and meiosis? So again, I'm just going to do a little bullet point up here for prophase. Oops. Have a little bit of an arrow here. And then I'm going to explain it here. This is about chromosomes condensing. Chromosome condensing. To prep them for division. Perfect. And then telophase. Telophase here is basically undoes prophase. adding back the nuclear envelope. I'm just going to move that down so it looks just a little bit prettier for y'all. All right. So basically that's all it's asking for. It's like, what's the similarities and differences? Bottom line is that telophase is the opposite of prophase. So whatever you're doing in prophase at first is going to be undone in telophase. It's like, thanks, buddy. You're neat. Last question. At what stage or stages does or do the most differences among mitosis, meiosis 1 and meiosis 2 occur? For what reasons do these differences exist? So the primary differences, like my bullet points, is basically, how's the best way I can explain this? It's basically the synapsis in prophase 1. That is the biggest difference. It's probably even the primary differences. I'm going to move that up and to the side. And then what you'll see here is that it is the primary differences because it allows homologous pairs to separate. To opposite poles and reduce the chromosome number at the end of meiosis 1, going down here, to half the original cell. And that is it, everyone. I keep forgetting I had more room over here. Good job, me. Yay. All right, everyone, that was it. Made it to the end. Probably a little bit earlier than what I expected. But yeah, hopefully this was helpful. Like I said, I will be available for tutoring starting at about 7:30 tonight. And I'm going to go all the way to 8:30. I could probably even push it to like 8:45. If you guys want to chat with me on the bio Discord, you're welcome to do so. And if you're listening to this recording right now on YouTube, you are more than welcome to ask questions to me on the bio Discord. I can also give out my email in the chat if you guys want that. You know, email me at my email. And if that doesn't work out, you can also book me on Penji. I have hours from like 9 to 10 tonight, but if they're taken, just email me and then I can give you a Zoom link and then we can chat there, because then I can share my screen like I am right now. I also have all of Dr. Reddy's old presentations, and I'm very familiar with how she asks questions on her exam. So hopefully if you come to tutoring, I can help you out. And if you don't want to come, that's okay. I'm wishing you all the best, and I hope everybody has a wonderful rest of their evening, and thanks for attending tonight's live stream.